Hi, my name is Jacob, and my project is on investigating environmental stress response genes in the grain crop sorghum using course-based undergraduate research labs. Many people are feeling stressed about the detrimental effects of climate change, but did you know that plants are experiencing stress too? Climate change induces environmental stress in plants, including exposure to excessive drought, abnormally hot and cold temperatures, and aggravating the accumulation of salts and other pollutants in soil. Climate change is expected to drive agricultural productivity declines, create food safety issues, disruptions in food distributions, and lower the nutrient quality of some crops, which will only exacerbate the already high level of food insecurity. Sorghum, the plant I've been working with, is a staple grain crop that is an important food source in many developing countries. Sorghum is generally considered resistant to drought stress, however, drought can still greatly reduce its productivity and nutritional quality. Through the use of molecular, genetic, and computational tools, sorghum genes that confer tolerance to drought and other environmental stresses can be identified. These genes could provide insight into breeding crops resistant to the adverse effects of climate change, making agriculture more efficient and helping to stave off food insecurity. So, the project began with growing several different genotypes of sorghum plants in the greenhouse, and after 3 weeks, plants were exposed to different environmental stresses, including drought, heat, cold, and salt stress. Following the stress exposure, plant tissue was collected and flash frozen to preserve its post-stress molecular state, essentially freezing it in the moment of gene expression it was in when responding to a given stress. I then took the frozen tissue and purified RNA from it. Using RNA will allow for the genes expressed in each plant and under each stress to be isolated. Next, using an enzyme called reverse transcriptase, the RNA was converted into cDNA, a type of DNA that only has coding genes. Finally, through intricate molecular biology techniques, the cDNA from sorghum was integrated into yeast cells, allowing the yeast cells to express those genes from the plants. Then, yeast cells with and without the genes of interest will be exposed to stress and control conditions. This approach will allow us to determine which genes, if any, contribute to environmental stress resistance. However, this work also extends to the classroom. Another major part of this project involves using this research to develop a course-based undergraduate lab experience where students will learn practical lab skills while also contributing to the discovery of novel stress response genes, immersing them in the day-to-day work of a professional scientist. This was a great opportunity for me to learn more about molecular biology, genetics, and research in general, and I'm deeply grateful for all the faculty and institutions that made this project possible. Thank you so much.